Welcome to a new episode of A Cellular Sensation. I'm Marie Elio, your host, and today we'll be staying on the surface of the body and looking at Australia's national cancer. Skin cancer is the uncontrolled, irregular growth of skin cells. There are three main types: melanoma, basal cell carcinoma, and squamous cell carcinoma. Let's have a look at the skin to really understand this. The skin has three main layers: the epidermis, or outermost layer of skin, the dermis, which contains hair follicles and blood and lymph vessels, and the hypodermis, mostly made up of subcutaneous fat. On the basal membrane of the epidermis sit the stratum basale cells. These cells are like stem cells, continually dividing to regenerate the skin, and this is where BCCs originate. BCCs may be light or bright pink in colour, appearing as a pearl-like lump or a scaly area, growing slowly over months or years. Above the basal cells are keratinocytes. Mutations in these cells are responsible for SCCs, which often appear as a thickened, red, crusted or scaly spot or lump. These are more common as you age, growing quickly over weeks or months. Amongst these cells are melanocytes. They produce a protein called melanin, which gives skin its characteristic range of colours. Mutations here cause melanoma, which can appear as a newer or existing spot that evolves over time and may have multiple colours. For the rest of this episode, we'll be focusing on melanoma, the most deadly of the three. We'll cross now to the epidemiology team to hear about the current climate of melanoma in Australia.
Today, the sky is looking clear for Australian melanoma. It is the fourth most common cancer diagnosed in Australia, and although it accounts for less than 5% of skin cancers, melanoma is known to cause more than 70% of skin cancer deaths. This equates to one person dying every 6 hours in Australia due to this cancer. Crikey! If we zoom in to different parts of Australia, we see that due to the differing climates of our big country, Queensland has a melanoma rate 3 times that of Victoria. Also, melanoma is less common among Indigenous Australians than non-Indigenous Australians. Back to the studio.
Now that we know how big a problem melanoma is, let's take a look at the science behind it. If we look back at the skin diagram from earlier in the program, we can see that melanocytes sit amongst other cells in the dermis. In healthy skin, when UV rays hit keratinocytes, these cells signal melanocytes to stimulate melanin production. This protein is secreted into nearby keratinocytes and acts as a natural sunscreen, protecting skin cells by scattering UVB rays. However, in melanoma, this process is damaged. The two greatest risk factors for melanoma are genetic susceptibility and high UV exposure. These factors can cause mutations in the DNA of melanocytes, leading to melanoma. There are two main types of mutations that can cause cancer. The first is a proto-oncogene activation. Proto-oncogenes are responsible for the promotion of cell division. When these genes are mutated, they are continuously switched on and cause rapid cell proliferation. An example of this in melanoma is the activation of the BRAF gene, which is found in 40 to 60% of melanoma cases. The second kind of mutation is loss of tumour suppressor gene activity. These genes are normally the brakes of cell division, inhibiting cell growth when there is damage to the DNA or division is unnecessary. Mutation allows uncontrolled growth even in the presence of damage. In melanoma, a common tumour suppressor gene mutation is to the CDKN2A gene. When uncontrolled cell division occurs, tumours form as cells pile up on each other. The piled melanocytes form dark or varying coloured tumours which grow horizontally through the epidermis and vertically, breaking through the basement membrane to invade the dermis. Here, tumour cells gain access to blood and lymph vessels for nutrients and waste excretion. Angiogenesis occurs and the tumour gains metastatic potential. Malignant melanocytes from the tumour can break off and metastasize in other parts of the body. Now understanding the science of melanoma, you might be wondering, how do I know if I've got it? Well, as usual, let's chat to an expert to find out what the disease looks like. Hi doctor, can you explain to us how melanoma is detected?
Sure thing. Melanoma is tricky. It doesn't usually have any symptoms except for the appearance of new skin spots or changes in existing moles. However, as clinicians, we use the ABCDE rule to help us diagnose a cancer. Each letter stands for something we look for in the skin group. Firstly, whether it's asymmetric, having an irregular shape. Then we look at the border, whether it's irregular or notched. Thirdly, we look at the colour. Growths often contain a number of different colours or shades. D stands for diameter, whether the spot is larger than the size of a pencil eraser. And finally, E stands for evolution. Melanomas usually change quite quickly.
Right, so what do we do if we notice one of these things?
You should definitely go to the doctor.
That's all we have for today. Catch us next time on A Cellular Sensation when we'll be diving into the digestive system.